Hello and welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and it's amazing that you're here today. And we will be talking about how drugs interact with anti-targets with Mark Murcko today. So good morning, Mark. It's a really pleasure to have you.
Thank you for the invitation.
And now, without further ado, I want to introduce our guest, Mark. So Mark doesn't need much of an introduction, so I'll try to be really quick. Mark is a senior lecturer in the Department of Biological Engineering at MIT and is also a principal at Disruptive Biomedical LLC. Mark received his PhD in organic chemistry at Yale and began his career at Merck, where he helped discover multiple clinical candidates, including dorzolamide, which is the carbonic anhydrase inhibitor for the treatment of glaucoma, and later became the first drug in pharmaceutical history to result from structure-based drug design. Mark is also a founding scientist at Vertex Pharmaceuticals, joining in 1990, where he advanced to Chief Technology Officer and chair of the SAB. And during his tenure, he contributed to more than a dozen clinical candidates and Vertex's first 7 marketed drugs. And since leaving Vertex in late 2011, Mark has helped ideate, launch, build, run, and advise more than 15 organizations working on diverse drug discovery modalities and technologies. Notably, he is the founder, board member, and from 2016 to 2018 was the founding CSO at Relay Therapeutics. He's a board member, strategic advisor, and from 2018 to 2021 was the founding CSO and chief innovation officer at DuPont Therapeutics. And he currently also serves on several additional corporate boards and SABs. Mark has directly contributed to 10 marketed drugs spanning glaucoma, HIV, HCV, cancer, and cystic fibrosis, and is the co-author on more than 90 publications and co-inventor on more than 50 issued patents. Among many notable awards, Mark was just inducted in the Medi Hall of Fame at the ACS meeting in Washington last week. So congratulations on the award again, Mark. He's also a past trustee of the Gordon Research Conferences and currently serves on the board of advisors of the Boston Museum of Science, the Possible Zone, which is a nonprofit that provides entrepreneurial and mentoring support for Boston high school students. And Mark also frequently participates in hikes to raise funds for charitable groups such as Life Science Cares. Mark, really an impressive CV. We're honored to have you here. So again, welcome to the show.
Thank you, Dennis. Good to be here.
Awesome. Mark, and in the good old drug hunter tradition, before we start with the talk, we always start with a few warm-up questions. So I wanted to ask you, what inspired you to start a career in drug discovery?
Well, it's really simple, actually. I'm a physical organic chemist by training, and about halfway through grad school, I realized that physical organic principles might actually be able to help drug discovery. And so the chance to do really cool science and to create medicines to help people, the combination of those two things seemed very exciting to me.
Yeah, super cool. Yeah, my advisor was also a physical organic chemist, so this is super cool story. What's your favorite molecule, Mark?
So I go back and forth on this. Actually, I'm going to give you a non-answer, because what I'm going to say is that every drug, it seems to me, almost like a miracle. It is so hard to make a drug that I don't have a single favorite molecule. I just love what we can learn from each and every example of a successful new medicine. And then a little bit less than that, you could say any molecule that's well crafted so that it enables you to test a clinical hypothesis. Because so often we don't understand the biology. And what we really need is we need tool compounds that enable us to address the biology challenge. So molecules that do that are also incredibly exciting.
I love that. I love that. Yeah, you could figure out so many new targets, and I think we have just tapped the surface only on targets at the moment. And I personally believe we're at kind of a breakthrough time in drug discovery. And yeah, we'll hopefully hear more about that in your talk as well. What are you most excited about today in drug discovery, Mark?
Well, there's a lot of things, okay, because I'm involved with a lot of different companies and a lot of technologies. But if I were to really boil it down for this audience, considering, all of us as chemists mostly and drug hunters, the fact that small molecules are not dead. Okay. A few years ago, people were saying, oh, there's no need for small molecules anymore. Well, I think we've gotten past that. I think we understand the tremendous value that small molecules bring. And partly that's because synthesis is getting to be more enabled. Um, it's now possible to make molecules that we really struggled to make before, or that were simply impossible. So I love that. I'm also really happy to see that as a community, we've finally gotten past all those very silly restrictive rules, about what a drug must be. We're much more open-minded now, I think, to new modalities, larger, more complex molecules. I think all of that restrictive thinking really was setting us back, and I'm very happy that we've gotten past that. So that, of course, relates also to new modalities, heterobifunctionals, and so forth, things we never would have considered 20 years ago. Um, and I also think that we're getting past the idea that all drugs have to be oral. I think the idea of being able to deliver a drug through a once a week injectable, for some patient populations, that's totally fine, as we've learned with all the GLP-1s. So thinking more deeply about the nature of how we get the drug to the patient. Sometimes it's oral, or sometimes it may be another route of administration, which is equally easy for the patient. And then the last thing I would say, and this comes back maybe to that 2018 paper that I wrote on what makes a great medicinal chemist. I've continued to think about this a lot. And I think we do have a kind of a playbook for the behaviors and the environment in which we want teams to be functioning, which together, I think they do dramatically increase your odds of success. Nothing guaranteed, but if we have the right behaviors and environment, I've seen this over and over again. I see teams doing better work and producing more. So these are some of the things that I'm excited about.
Amazing. Thank you so much for sharing, Mark. And if you could go back to the beginning of your career and give yourself some career advice, what would you say to your younger self, and maybe to the younger folks in the audience here today?
So the first thing I would say, building on the point I just made about behaviors and environment, when I was early in my career, to me, it was all about the science, the technology. I did not pay any attention to these, so-called softer, squishier factors. It wasn't that I was disdainful of them. It was that I just didn't pay any attention to them. But these days, I would say half or more of the time that I spend is on these cultural, social kinds of factors, how teams are doing their work. So that's one thing that I would say to early career folks, is really pay attention to that, as well as, of course, the quality of the science. And then the other thing I would say is a lot of experts, you want to listen to them, but you don't automatically want to believe them. When I was early in my career, I was very deferential to experts. And so I'd be in a meeting and someone would say, oh, we can't do this because, or the only way that this will work is. And I would say, oh, okay, this person is 20 years older than me. They have all this experience. They sound like they know what they're talking about. I'm going to believe them. I don't do that anymore. And I don't want anybody to do that when I say anything either, because yes, experience is incredibly valuable. I'm not disputing that. But I think that because what we do is so hard, so challenging, we want to listen to the experts, but also really try to deeply understand what leads an expert to their point of view, and to question it and challenge it, obviously in a collegial, polite way, but to really understand it and not just say, oh, that must be the answer. So those are a couple of things I would suggest to my much, much earlier self.
I love that. No, that's the getting kind of really to the root cause of something, and really to understand why they believe it, because probably they had a certain set of experiences that led them to believe that, but now maybe something challenges that viewpoint. I love that, really.
Yeah.
Well, last but not least, Mark, what do you like most about Drug Hunter?
Well, so a big part of how I think about things is I do try to learn from history. I try to learn from all the examples around me, what works and doesn't work. And so Drug Hunter is just amazing because, it's you guys do a really good job and I see Lou on the screen too, and Lou is part of this, of course, as well. You guys collectively, all of you on the team, you do a really good job of gathering and organizing and summarizing what's going on in the field. And I think that enables all of us to learn these lessons, which can help all of us. So that's a real service, and it's great that you guys exist. And I wish somebody had that. I wish somebody else had done this 30 years ago, but it's great that you guys are doing it now.
Awesome. Thank you so much. That's a tremendous compliment. Thank you so much, Mark. And I want to show, before we get started with Mark's presentation, I want to show people, a quick snapshot of the Drug Hunter platform and what you can do with it. One quick example of what you can do with it. And today I want to show you our patents. So our patent search function. You can click here on patents, and you immediately get a list sorted by the month, the target, the therapeutic area, the originator. And what we also do is we pull out some highly important, high value information from those patents. So that will be like how many compounds are in this patent? Is there any like interesting biochemical data and interesting DMPK data or efficacy data? And you can search even here by, let's say, the annotation. So you can search, for example, if you want to type brain, if you need, for example, let's say you're looking for a brain penetrant compound. You can say, for example, contains brain, KP, or CNS. And then immediately here, find high value patents where you can read a quick summary of the patent, get pointed to the most important compound in the patent, and really get great information about this altogether. So here we see dew point therapeutics. So Mark, of course, is intimately familiar with. We can also additionally sort, let's say we want brain penetration in neurology. We can narrow it down further to neurology if we're interested in what targets are people looking at in neurology, where there are patents with brain penetrance data. We can easily sort this by target here and really quickly get an idea of these targets that people are working on, the companies that are working on those targets, and again, the most important data from those patents. You can also click here on patent highlights, and always before the next month ends. So here we have July patent highlights that come out before the end of August. And here we highlight some interesting chemical structures associated with data. So here this interesting phosphorus-based prodrug, for example, or Mark highlighted the GLP-1 space a little bit earlier. And here you can see like patents in this space. We talk a little bit about the importance of the target, the competitive landscape, and how those compounds in this patent here differentiate from the existing chemical matter, and much, much more. So with that, I will hand it over to Mark for his presentation on how we can solve the hardest problems in drug discovery.
Well, thank you, Dennis. It's great for the invitation, because I'd like to describe a project that I've been working on for several years that addresses a huge challenge that we all face, and you could even call this a disruptive idea. I hope you'll find it interesting. I've talked about this a tiny bit in a few other recent talks, but this talk today is the first time that I'm presenting the whole concept, because I really think this is the right audience, and I'm very much looking forward to getting feedback from you all. And I'd like to begin by telling a few stories that illustrate why today's topic is so important for all of us. My very first project at Merck, straight out of grad school, was the design of carbonic anhydrase inhibitors, as Dennis mentioned, and that was for the treatment of glaucoma. The target was structurally enabled, which at that time was pretty unusual, and there were already nanomolar inhibitors. But the problem we faced was topical delivery via an eye drop across the cornea, which is a very complex structure with alternating hydrophobic and hydrophilic layers. And this required carefully balancing the physical and the PK properties so that we could optimize the delivery of that drug via the eye drop formulation. And of course, the X-ray data we had, very useful for helping us maintain potency, but it was of no use in helping us to understand the subtle changes in the ADME properties that we were seeing from one molecule to the next. So Another example, this was when I was doing some consulting, this is about 10, maybe 12 years ago. It was a project where a team at a large company was having trouble separating on-target activity from efflux. The compounds were PGP substrates, and the SAR tracked very, very closely. And the team was working entirely in the dark. They had no atomic level understanding of why these molecules were PGP substrates, and ultimately they gave up the chemical series. And then a few years later, at Relay, one of the very first projects we had, we faced a herd challenge, and it took considerable time and effort to sort it out. And it was only after we had synthesized and tested hundreds of compounds that we could build a decent predictive model. So we did solve the problem, but the fact that it took so long and it was painful, required so much experiment, that really bugged me. And then a more recent example at DuPont, a team at DuPont had been dealing with a time-dependent inhibition problem, and the molecular cause was not at all obvious at the beginning. Happily, the team did sort it out, but again, it took a very long time. So these kinds of stories and hundreds of others that I've heard from colleagues around the world, all of these lessons have taught me something that I'm guessing is already well understood to just about everybody in the audience, and that is that affinity against the target of interest, of course that's important, and it's personally, with my background in physical organic chemistry, it's something I love thinking about. But all of these stories, they indicate that affinity is obviously not the only thing that matters. And in the end, a drug only works because you can deliver enough of it to the right tissue. It hangs around long enough to achieve the desired pharmacologic effect. And it, of course, has to have a good therapeutic window. That's what makes a great drug. And there are so many factors that have to be balanced. This series of graphs, it comes from a very useful retrospective paper that describes 25 years of projects at Novartis. It shows how the optimization of each of these many properties, each property requires tremendous effort and time, and it's certainly not a linear process. And that's why drug discovery is often called a game of whack-a-mole, because multi-parameter optimization, that's the challenge we face. So our common goal, all of us face this, is we want to improve our ability to create molecules that have the right mix of properties. And as a field, all of us med chemists, we've developed all kinds of ways to analyze our ADME and our tox properties. And we talk about shape, lipophilicity, charge, conformational flexibility, hydrogen bonding. And we're always developing better ways of thinking about those properties, either by experimentally determining them or calculating them. And then we use those values to create predictive models that sometimes work quite well. I use these. I enjoy this as much as everybody else because they do sometimes help us to explain our data. But notice that this approach is almost entirely centered on the drug molecule, because changes in the ADME and tox properties across our series, we think of how it's affected by some change in biodistribution, which we don't really understand. It's kind of vague. Perhaps it's a bulk property effect, lipophilicity, charge, dielectric, ionic strength. Maybe it's a non-specific interaction with phospholipids, generally speaking, or with some collection of proteins. We rarely think explicitly about how changing the properties of our drug affects, at a molecular way, the way we are interacting with all of the other proteins in the body. We think about that a little, but I don't think it's front of mind. We're mostly focusing on the properties of the drug molecule rather than all the things in the body that the drug may be interacting with. Now, here's another example of that. Percent F, it does vary from one species to another. And I know sometimes there can be nice correlations where the rat and the dog and the monkey PK all look the same. That's great when it happens, but it doesn't have to be true. And in fact, there are plenty of reasons to expect differences from one species to another, and those are listed in the blue box. And of course, most of these are clearly related to ADME properties. And by the way, I want to acknowledge an amazing pharmacokineticist, Ian Martin, who I had the pleasure of working with at Relay. He's been a wonderful guide for my recent forays into the subtleties of ADME. And Ian, by the way, is transitioning to a full-time consulting role. So if you're looking for expert help, seek this guy out. He's amazing. Now, some people will argue that since artificial intelligence is here, all these problems will magically vanish. Unfortunately, that's not true. The best way to improve machine learning models is to feed them more data and higher quality data. It really does not matter how powerful in principle a co-folding or generative or other machine learning method might be, because all of these methods have to be trained and validated. And that's especially true when you think about the sheer complexity of these ADME and tox proteins. And I'll talk a little bit more about that later. Just for now, I'll mention they're multi-domain, they're highly mobile and flexible, they have multiple functions. And of course, different drugs will bind in different poses at different sites. And then, of course, the drug space is many orders of magnitude larger than the protein space. As hard as the protein folding problem was, figuring out how small molecule drugs interact with proteins is many-fold more difficult. So we simply need much more experimental data. And without that data, there's a severe limit to the value that we can achieve from machine learning methods. Now, of course, as I've already said a few times, it's easy to focus on binding affinity, optimizing intermolecular interactions, strain energy, all of those things, using FEP. I ran my first FEP calculations in 1989. Okay, so, and of course, I still see great value in thinking about trying to measure and improve binding affinity. That's all true. But at the same time, my argument today is that to maximize impact, structural biologists, assay developers, biophysicists, molecular modelers really have to focus much more on ADME and tox. And by the way, this is no different conceptually than any other structure-based design situation. We always work on anti-targets. We always want to design away from them. This is the same concept, but it's on steroids because it's not a single anti-target. It's the fact that there's 1,000 of these proteins that we have to deal with. And by the way, as a quick aside, structural biologists have been solving protein structures of ADME targets for ages. I think the first bacterial P450 was about 40 years ago. And the idea of focusing in a more systematic way on these ADME and tox proteins, that idea has been around for a long time. For example, here's a paper from 14 years ago from Bayer. Alex and his coworkers, they were clearly way ahead of their time in pointing out the importance of these anti-targets. So now let's define our terms a little bit more carefully. So what is this collection of proteins that we're worrying about? Well, there's about 1,000, maybe more, proteins that are involved with metabolism, transport, efflux, all the other ADME mechanisms. And in addition, there are, of course, so many third rail targets that we know are dangerous to hit because they'll lead to toxicity. So together, collectively, we can refer to this set of undesirable proteins as the avoid-ome. These are the things that we simply do not want our drugs to interact with. And we all know these targets. We deal with them every day. Proteins involved in metabolism, drug transport, extracellular proteins like serum albumin, proteins like PXR that affect xenobiotic transcriptional response, or any of the large number of proteins that can lead to toxicity. I guess hERG would be a poster child for that. So we know what these proteins are. And then you could ask, well, of the 1,000, which ones really are the ones we care about the most? Well, thinking about each of us has our own personal experience, and by reviewing the literature, talking with colleagues, chemists, pharmacokineticists, toxicologists, one might attempt to map out a rough prioritization of the avoid-ome. You might start, for example, with the targets in the dark gray box on the left, and then add the targets in the medium gray box in the middle, and then the targets in the light gray box on the right. Now that's an attempt at prioritization. You might do it differently. It's a judgment call. I've run this list by a lot of people. Mostly people seem to agree with it. There's always new suggestions coming in, and I'm actually very interested in any thoughts that anybody in the audience has on whether you might remove some targets from this particular list or add others. It's got to be a communal understanding of what really are the most important targets to start with. And yes, of course, this collection, or any collection on the order of this size, this is about 65 proteins, we know it won't cover every possible case. I think it covers most of the ones that we tend to run into in our daily lives, but there will certainly be some chemical series that are interacting with a different avoid-ome target. And as we learn about those, we can add them to the list and pick them off one by one. Now, so I've just described this idea to you. I have learned from bitter experience over many years that before I ever go after trying to systematically tackle anything new and disruptive, it's always useful. It makes a ton of sense to try to poke holes in that idea. I think it was Richard Feynman who said that when it comes to your own ideas, you are the easiest person to fool. So I am a big fan of what some people call pre-mortems, which is a way to try to anticipate all the things that could go wrong. So I've run this idea past many colleagues and had many great conversations about it. A number of objections could be raised. And so I think it's useful to walk through these just for a minute to give everybody a feel for whether we agree that, that these are serious concerns that would make the whole idea just a bad thing to pursue, or whether, in fact, we think it's still a good idea. So first of all, we need drugs to get out of the body. So we like the fact that drugs are transported and metabolized. That's of course true, but I would argue, and I think you'll all agree, most of the time we have the opposite problem. We're interacting with too many of these targets or too strongly. I think most of the time we want a longer half-life or a higher free fraction, or we want the drug to go to the specific tissue that we're interested in treating and concentrate there. Those are the real challenges that we usually face. A second objection is sometimes avoid-ome targets are actually the targets we want to inhibit. Here are a number of examples, and that's absolutely true. And those are great. Those are specific cases, but these are, as I say, a handful of special cases. And in other more typical kinds of situations, you would not want to interact with these targets, or at the very least, you would want to understand how your molecules are interacting with them so you could adjust the activity as needed. Now, a little bit related to that is sometimes we try to exploit avoid-ome targets in very specific ways, such as binding to serum albumin to extend half-life. Or maybe we're trying to create a prodrug or a soft drug. Well, these are all examples of cases where we try to use avoid-ome targets. And so to me, it's just another example of how it's valuable if we can understand in detail at a molecular level how our drugs are interacting with the avoid-ome, because that will enable us to more effectively utilize these anti-targets for our own good purposes. Okay, now there's a whole range of objections that a structural biologist might raise. A crystallographer might say, hey, I'm expected to solve protein ligand structures. That's what my team wants me to do. Or they might say, well, potency, that's important, and it's something I can help with. So shouldn't I focus on that? That's where I can make my biggest contribution. So I think these are both reasonable points. I don't quite agree with them. I think the right way to think about it is that, of course, solving liganded structures is incredibly valuable, but only up to a point. I've seen over and over again that after a team is well established and has many protein ligand structures for their chemical series, the 27th ligand structure, it usually provides diminishing returns. Yes, there could be exceptions. That 27th molecule might have some very strange SAR, it might be an activity cliff, and then you have a specific reason for wanting that structure. I get it. But other than those special cases, I think experience would bear this out. I think once you get to about 20, maybe 30, but more likely 20 ligand structures in a single chemical series, I think you're probably not going to learn all that much from the next structure. Now, on the second objection about potency, my answer, I've already talked about this, is yeah, potency is great. I love it. Keep trying to make molecules that are more potent. No argument at all. But I would argue that past a certain point, additional affinity will not be as valuable as focusing on all the other factors that make a great drug. Remember, it's a multi-parameter optimization problem. So in short, in both of these answers, what I'm arguing is that focusing primarily on ADME and tox to help a team make a better molecule, that would in fact optimize the value that a structural biologist is bringing to the table. And I want to say just a tiny bit more about this topic, because this endeavor, it's far too large for any single lab. We're talking about many, many proteins, and they're complex proteins. So it would be hugely beneficial for the entire scientific community to share in this task. And think about the transformational impact we could all have together if we, over, say, the next 5 to 10 years, if we collectively tackled this challenge. And I think this effort is totally aligned with the role of structural biology in industry. And I just saw some little hearts go up the screen. I guess that means somebody liked what I just said. So whoever you are, thank you. I also think that there are multiple benefits. There's more hearts now. I also think there are multiple benefits for the structural biologists themselves. Obviously, this is the most critical impact because this is the challenge that project teams are wrestling with. This is what prevents us from getting our drug candidates across the finish line, right? So if a structural biologist can help with that by unraveling the mysteries of the avoid-ome, the chemists will absolutely love you. A second example is it expands the playing field. What if right now a project team is working on a target that can't be structurally enabled for whatever technical reasons, or maybe it's a phenotypic project where the target is not known? Well, in those kinds of cases, right now, structural biologists can't do anything. But instead, if you focused on the avoid-ome, the project teams would greatly benefit from the structural insights on how those chemical series are engaging the avoid-ome. So you could be much more useful by expanding the set of targets and projects that you could be working on. And then, of course, on top of that, these are very cool proteins, just from a fun science standpoint. These are multi-domain, multifunctional, complex, mobile. So these are great targets, just for the intellectual joy of it. And then, of course, all of this work, I would argue, is pre-competitive and it's highly publishable. I think there's a lot of reasons why this would be a wonderful thing to see more of. Now, another objection that I've heard a few times is some people will argue that buried in the literature or locked away in the vaults of big pharma is all the data that we could possibly need to help us understand the avoid-ome. It's a cool idea, but I think there's actually very little evidence for this. Of course, I fully agree. There is some pre-existing data that I would dearly love to see. Maybe certain assay protocols of sets of results on certain screens or structures of certain avoid-ome targets. No doubt there is some data out there and I wish it would be shared. But I think for the most part, it's safe to say that this would be sparse and inconsistent, difficult to combine, totally inadequate for our purposes. It's not enough. It might be helpful, and I hope it happens, but it's not like magically all the data we ever need is already out there. And again, remember just how complex these proteins are. So I think instead, what we need is a great deal of high quality new data on well-chosen targets and compounds done systematically, consistently. And it requires a careful mix of useful compounds, well-controlled assays, thoughtful machine learning, carefully chosen structures, and all of that will have to be done on an unprecedented scale in order to go fast enough to really have an impact. Okay, now, just to humor me, I hope you will, let's say that I have addressed these objections to a reasonable degree, and that we're all willing to consider the idea that, yes, we should be embracing the avoid-ome. So how would we actually go about that? Well, we have to, again, embrace just how fiendishly complex these proteins are. We need multiple snapshots of these structures and with diverse ligands, and we need to explore the range of motion of these proteins, and we need to do that both experimentally and with molecular dynamics. We need affinity data as well as functional activity data. And all of this will require at least an order of magnitude reduction in the cost of screening. And we'll need to be able to synthesize the molecules we need, the right compounds to flesh out SAR, understand activity cliffs. So of course, a big part of this is structure, but it's all these other things as well. It all has to be put together. And so if we wanted to build a global approach to tackling the avoid-ome, well, what would we like it to be? What do we want it to look like? You would want to be generating 3D structures and assays and data sets and models. And you would want all of the data to be in the public domain. And you'd want to be using the best possible machine learning to prioritize compounds and to continually learn. And you'd want the structural biology, of course, of all of these targets, but also with specific ligands bound, so you could understand the outliers and the activity cliffs. And then you'd want to demonstrate how all of this data, used in the right ways, would lead to better predictive models. And you'd want to use blind challenges as a way to get ground truth, to really understand what's working and to stimulate more and faster participation and discussion and collaboration. So all of this sounds great. And the good news is that it already exists. So I'd like to introduce the Open ADMET Consortium. And this is an organization with a very specific mission. The goal is to build open predictive models for safety and tox and PK for small molecules. And the goal, of course, of that is to improve our ability to more effectively discover new medicines. And we've already been awarded grants totaling tens of millions of dollars from the organizations on the right. The three principal investigators are at the bottom: Jamie from UCSF, who focuses on structure, John from Memorial Sloan Kettering, and from the Open Molecular Software Foundation. He, of course, works on models. And Sri from Octant, who has a team developing dozens of high-throughput, high-quality assays. And Pat and I helped to flesh out the idea, and we serve as advisors. And there are many other investigators who are also already involved in this. And an important benefit of Open ADMET, the way we see it, is that we'll be exploring these diverse new kinds of assays. And from that, we'll be learning a great deal about which kinds of information actually have the greatest impact, which are the most helpful for us in drug discovery. And so to achieve that new understanding, we will need consistent data across large and diverse areas of chemical space, across large numbers of targets. So that in turn means we need to be able to make and inexpensively test these compounds. And so for this purpose, the Open ADMET group is taking advantage of the fact that Octant already has a high-throughput nanoscale synthesis capability, and they have a direct-to-biology assay platform. And so that enables Octant to synthesize thousands of compounds and then just take them straight into the ADME assays that they're developing. And to do that, of course, they've put as you'd expect, a diverse team of people on it: analytical chemists, data scientists, biologists, synthetic chemists. And it's early days, but there's been some really nice progress already. This is one example where Octant was able to build a custom PXR assay, which has much better signal to noise than the commercial assays. It's scalable to 1536 well plates, and it's already about an order of magnitude cheaper. In the middle, you see the results for a set of about 1,000 diverse compounds. And as far as we can tell, this is a real advance on what's publicly available for PXR. And of course, this data will be released to the community, and there will be many more examples of different assays like this coming in the near term. And then once you have that PXR assay in place, then the whole Open ADMET team kicks in, and the goal is to do active learning, and that involves building machine learning models, sampling where the predictions are not very certain or are flat out wrong, and then going and generate more data around those compounds and then analogs of them to get more data that can be fed back into the models to iteratively update them and learn. And then, of course, you would want to structurally do this as well. You want the highest resolution PXR structures available, and Jamie has developed that, and that enables you to then go after the information-rich compounds, the ones that you get wrong, because when you get the structures of those, you get all kinds of new insights, which then enable you to further refine the predictive models. And on this topic of predictive models, it's an open question of just how far we can go. How accurate will the predictions be on targets that are as complex as these sorts of avoid-ome targets? So the folks at the Open Molecular Software Foundation, that's what they are working on. And you can look at, for example, all the known inhibitors of a target, in this example, CYP3A4, and then, like I just mentioned in the example of PXR, where you're trying to do active learning, you can choose novel compounds that are not well covered by the current predictive models, or compounds that are wrong. And then you can continually update the models by generating that new data and rebuilding the models. And that helps you to assess the quality and the robustness. And the overall process at the Open AdMet Foundation, because we'll be generating such a very rich data set across so many targets, that also is really valuable because it enables us to address some very fundamental questions about how to use machine learning itself, how to fine-tune a model. Very hard problem to answer. When really can you do machine learning on a multi-parameter optimization sort of way? That's very hard to know right now. And so the beauty of this is what we learn on this project, these lessons will be transferable to all aspects of drug discovery, far beyond the avoid-ome. And at the same time, also remember, all the experimental data will be in the public domain. And so that way everybody can take a shot at analyzing it. Now, structure, of course, plays a number of important roles. So first of all, recall that chemical space map that I just showed you on the CYP3A4 known ligands. Well, we can think about how can we extend the quality of the methods by finding new binding sites. And so we can seed new regions of chemical space by performing X-ray fragment screens, which Jamie is all set up to do and has done it. And these new structures that bind in different places, those can be used to improve the performance of, for example, co-folding methods, because the co-folding methods will now have far more information about different diverse places on the surface of a protein where molecules can bind. And then a second example, structural biology, I think I've already mentioned this, can help us look for outliers, because those are the ones where you learn the most. And so you try to identify the outliers and then go do structural biology on those, because that gives you new insights that help you to refine the models. These are the most valuable information-rich compounds to study, because you get the structural basis for why the prediction was wrong in the first place. And along the same lines, when you get those new structures, you sometimes find they are binding in a different way. You get unanticipated conformational changes in the ligand or the protein. You're making some kinds of new interactions, and all of that is hugely enabling for free energy perturbation. Because often when free energy perturbation fails, it's because you're starting with a crystal structure which is not quite right for that chemical series. And so the more we know about the structural variation that's possible within any given protein, the better job we can do of finding a more suitable starting point for the FEP. And an example of this was shown in a recent paper on hERG that the Schrodinger folks published. So the last thing I want to mention is that blind challenges are a very important part of how we're thinking about this, because that gives you ground truth. That lets you assess whether the field has actually moved forward. And we recently conducted a blind challenge, and that one was in partnership with the ASAP Antiviral Center. And in that case, the competitors had to predict three separate kinds of data. They had to predict on ligand poses, ligand potency, and also a set of ADME properties. And the challenge in this case happened to be won by a startup called Inductive Bio, and they were using graphical neural network and some other tricks. And we've recently posted a preprint with lessons that came out of this overall blind challenge, and we will be doing this regularly to really make sure that these methods are working. So there's all kinds of ways to contribute to this effort. You could share your data for blind challenges. You could encourage your teams to participate. You could put structures into the PDB. You could encourage your company to join an industrial consortium that we will form to help guide our efforts. There's a lot of things to do here. So I hope that it's clear that understanding the avoid-ome, this is a very powerful way to lift all boats, to make all of us more effective. That's going to enable us to bring more drugs to patients more quickly. And in the end, that's the only thing I care about. That's what this is for. So the focus is to start with experiments on a massive scale. We simply need tons more data. And then, of course, as the data appears, new insights will emerge. We'll be able to build far better predictive models, and the data is public. So researchers around the world will definitely be playing a critical role in developing these insights and models. And this is a huge effort. Everybody can contribute in all the various ways that I just described: solving structures, developing models, running models, synthesizing compounds, contributing funding. So I hope that it will be possible to work together with many of you in the coming years on this. And I'm very interested in hearing your thoughts today about this whole initiative. So thank you very much.
Thank you so much, Mark, for this very thought-provoking talk today. And we already have a lot of questions in the audience, which is great, which is exactly what you wanted. So I think let's start with the first question from Rose Host here. So let's say, people, and I'm sure this is actually the case, people will walk away inspired here today, and they will want to start on their project to collect data on the avoid-ome. Where, in which stage of drug discovery would you recommend to start collecting data on more globally on the avoid-ome? Is this already in the hit to lead phase or in the lead optimization phase? Like, when should people start?
I pay attention to ADME properties when I'm looking at hits that come out of screening. So to me, I would say hit to lead. Back long ago at Vertex, when we were setting up the new project team, the thing that we insisted on was that we would never call something a validated hit unless we had some inkling of PK data on it, because we didn't care about anything that didn't show that. So I'm a fan of doing things early. Now, I know you can overdo that, but I tend towards earlier rather than later.
Yeah, that makes total sense. And then, yeah, next question from Yoshi, and he's mentioning PXR specifically because you brought this example, but similar things are true, I guess, for HERC and other avoid-omes. So PXR is a highly flexible protein, and there's multiple pockets for small molecule binding. And there are, of course, X-ray co-crystal structures known for PXR binders. But Yoshi is wondering, do you think xenobiotic receptors like PXR are good candidates for machine learning and predictive models? It's like, I think these are multiple binding pockets, I think he's wondering.
So I think it's a really good point. One of the reasons we started with PXR is because there is some structural data, there is some information. And so the idea was, let's start with what's known and then ask, how can we extend it? So imagine that you get much more information structurally about how different series are binding to PXR. And what if you then take those hits and you build libraries of close analogs to generate that SAR? So if you do this in a very systematic way, and like I said a couple times, you're constantly looking for the outliers. You're constantly looking for the things that the models can't predict. And those are the ones that go back into structural biology and other computational methods as well. So it's a matter of iteratively continuing to learn. And will it be perfect? It'll never be perfect. But if you can get to the point where in most cases you have a far better understanding than we do today about how to proceed, right? If the assays are fast and cheap and you could run, let's just say, all 65 assays for $1 a well on pretty much any compound you make, think about how that would change the way you think about doing drug discovery, And then the models will continually get better. Every new piece of data gets fed back in.
Yeah. That makes total sense. And I think one of the challenges is to probably miniaturize all the assays. And I guess you've done kind of the first steps. So one of the questions in the audience here from Jason Pettis is, how do you scale a larger number of anti-targets without an explosion of costs initially? Because right now, I think, I guess we still have like high cost per compound for those anti-targets. So, how do you get started on that one?
Yeah. Well, I think what we want to do is in the next few years, we want to be able to produce as many of these assays as we can. And we want those to become widely available because then anyone can use them. So I think that's a partial answer. I think right now, I completely agree with the question. It's very hard to know what to do. Usually you don't even know what anti-target, what avoid-ome target you're hitting. You know that your drug is rapidly metabolized. You might assume it's a CYP, but you don't really know. And so part of this is about going from that situation to one where you can, precisely determine on a molecular level what's going on. And the sooner you know that, the better job you can do of then building that knowledge into your design strategy. You see?
Yeah, that makes sense. Well, also, in addition to like, let's say these avoid-ome targets where we like have a binding event, maybe to ligand protein binding event, is the AdMed consortium or the Open AdMed also looking for models to predict solubility and permeability? That's another question we have in the
Yes,
audience.
absolutely. That's in the work plan for sure. It's not just specific avoid-ome targets. That's the majority of the work, but there's also a number of, what do you want to call them, bulk properties assays like solubility, log D, and so forth. Yes, that's part of it too.
Awesome. That makes sense.
Because we
Uh,
want those assays to be cheap as well.
of
And right,
course, yeah.
right now, unfortunately, they're quite expensive. So what if we could measure log D much more cheaply or cellular permeability, for much less cost than MDCK currently is? That'd be useful.
Oh yeah, it'd be super useful. I mean, especially we were just talking about brain penetrance, like efflux. If we could predict efflux, right, that would be very very powerful as well, and potentially could predict brain penetrant compounds before even making them. Kurt Ritter is wondering, how will covalent binding compounds be thought about?
Well, I think he's raising a very good point, that in some cases, it's not just a matter of understanding the affinity to the target, but the kinetics. And I would argue that knowing the structures of how the covalent molecules are binding to the protein, that itself is quite useful. Right now, often we don't know that. So you have a covalent molecule, and let's say it's binding to an avoid-ome target. Once you know that, you don't automatically know where it's binding on the avoid-ome target. So there, I think having that structural insight into the pose, the binding pose, and the specific covalent bond that's being formed, I think those are also incredibly helpful to have. Now, some of that may not transfer as well because, of course, there's more complexity in general with covalency. Fully understand that. But I think if you had a set of a number of diverse covalent ligands against a particular target, and they were binding in different ways, and you had all that structural information, that would be far better than what we have now.
Yeah, that also makes sense. Well, the next question is more of a statement that's asking for your thoughts on the statement. So it's from Govindian Subramanian. Um, while an increased data set size or volume may improve on the predictive capabilities of an AI or machine learning model, I believe that the scarcity of proper description is the limiting factor. The field has been working with the same set of descriptors and expecting answers for all endpoints being investigated. He'd appreciate your thoughts on that one.
I actually, in some of my MIT classes, I have a slide on this. There's three things that matter. There's the data representation, which is what he's talking about, he or she. And then the third is the algorithms that you use. I think that the most important by far is the data. And then comes the molecular representation, as the question is suggesting. And then finally, the algorithm. I think at the end of the day, I don't want to say it's a commodity, but I think once you have the data and you have a molecular representation, there are many ways that then you can try applying machine learning to get insights from that. So I completely agree with the questioner that having better ways of representing molecules, not just smile strings, I 100% agree that that would be very, very useful. I don't agree that it's the most important thing. I think the data is the most important thing, but we could quibble over that. They're both useful.
Yeah, that makes total sense. That makes total sense. Yeah, well, we talked a little bit, or a lot actually, about the ATME. One, I want to touch a little bit on tox as well, and that's a question in the audience here as well. So it's from an anonymous attendee. A big problem is idiosyncratic drug-induced liver injury. There's also polymorphism
Yep.
in the expression in the different expression of humans. So I just wanted to like kind of ask, how do you think about those kinds of tox challenges
Yeah.
that oftentimes are identified much later, often in clinical phases
Yeah.
only
I
today?
I agree that that's a big issue. I think there may be some ways in which what we do will be helpful, but honestly, I think that's a separate kind of challenge. If
Hmm.
you don't see a problem until phase 3, okay, until you've got 10,000 patients in the clinical trial and there's 3 people who suffer acute liver injury because of some oddball situation, I don't think we're able to do a good job of addressing that problem. So I think, could it be that by knowing something about, for example, if your drug is interacting with glutathiones, could you gain some insight earlier into a potential for an interaction that could lead to a DILI? Yes, that I believe, but specifically idiosyncratic tox that's very, very rare and doesn't show up until many, many patients have seen the drug. I don't know that we're going to address that problem, at least not directly.
Yeah, no, that makes complete and total sense. Yeah. We have another question here from Michael Bradley. So thinking across multiple disease areas, but harnessing publicly available target discovery data from oncology, for example, from DepMap, you were asking like what other people think should be included in the Avoid-ome. So he's thinking about, what do you think about expanding the Avoid-ome to include a list of common essential proteins that seem to kill the vast majority of cell lines when they are genetically disrupted?
Oh, that's an interesting idea. I wonder. I'll think about that more. And thank you, Michael. Good to hear from you. I know Michael. He's a thoughtful guy. So it's a great question. I'll give it some more thought. I wonder what the overlap will be between those kinds of targets and the known tox targets, the third rail sorts of targets that we tend to worry about now already. I wonder if those sets are overlapping at all. I don't know the answer to that. So thank you. I'll think about that.
Yeah, sounds good. I think, yeah, we're almost at the top of the hour. We have a couple of more questions. I just want to maybe ask one more, if that's okay, Mark.
As many as you want.
Okay, sounds good. Cool. So this one is from Zachary Stein. He's asking, do you think that a traditional DEL campaign would be a useful or powerful tool here in identifying high affinity ligands to various avoidome targets? Or would the limitations in affinity selection design be too challenging?
That's a deep question because, as I'm sure a lot of people on the call know, there are a number of companies that are using DELs to try to pan for hits more broadly. And then there's the usual question about whether you believe that data or whether, directly from the DEL screen, or whether you need to clip off the tag and resynthesize the molecule and so forth. It's a good question. I think what it might be useful for is identifying novel pockets. That might be the best use of it. It'd be very interesting to compare what you get from a DEL screen to, for example, a fragment screen. I wonder if you'd get the same information about novel binding pockets that you're able to access on a given avoidome target. It's a good question. I'll think about that some more.
Sounds good. Well, Mark, with that, it remains to thank you for a very thoughtful presentation and sticking around here to answer the audience questions. Thank you for the audience as well for very engaging, thoughtful questions. Already appreciate that, of course.
And if I could just say,
Yeah, Mark.
anybody on this call, I would love to hear from you. Get in touch. If you have follow-up thoughts on this, send me an email. I'm easy to find. Love to hear what anybody is thinking about this.
Sounds great. And Mark, we can also promise to share. There's, I think, a couple of more remaining questions. We can share those by email with you as well.
Perfect.
Sounds good. So yeah, we will see each other next week here then. Hopefully, if you dial in next week, we, as Mark pointed out, not all drugs have to be oral, right? Next week, we're going to talk about long-acting injectables, lessons in long-acting injectables with Jean-Baptiste Langlois and Thomas Ulrich from Novartis. And they will talk about lessons in the discovery of the long-acting injectable TAC1 inhibitor from Novartis. So until then, have a wonderful rest of your week. And thank you again, Mark, for a great presentation.
Thank you.
All right. Take care, everyone. Bye now.